My name is Victor Lin, and I'm affiliated with the Department of Pathology and Cell Biology. And I'm also the manager who manage the Cancer Center genetic modified mouse model share resource. So today I'm going to present 3 new mouse models that we generate in the past year during the pandemic for the SARS-CoV-2 and COVID-19 research. So in the late of 2002, SARS emerged as like, it's not a pandemic, but it emerged for a couple months, then it went away. And in 2003, actually, Dr. Cho and for Zhen's group at Harvard University presented a paper in Nature to validate that actually the human ACE2 is the real receptor for SARS-CoV. And the human ACE2, in this paper, they link a C9 tag to the human ACE2 C terminals. And then they have shown that this fusion protein can mediate the infection of SARS-CoV in the viral cell. So it basically, this paper confirms that the human ACE2 is the true receptor for SARS-CoV. And Dr. Cho's group, actually, they deposited this cDNA, the human ACE2 with the C9 tag at its C terminal. They deposited this cDNA to the edge, and it's available to the general public. And so far, there are 3 animal models available for the COVID-19 research from Jackson Lab. And the first one is the K18 HACE2 transgenic mouse line. And in this transgenic mouse line, the human ACE2 is driven by the human K18 promoter. So the human ACE2 will be expressed in the epithelium cell. And the other 2 mouse line is a humanized HACE2 mouse line. The model 1 is they through the gene targeting strategy, they replace the endogenous mouse ACE2 with human ACE2. And the difference between the model 1 and the model 2 is actually model 2 contains a little bit more 3' UTR regulatory region. So you can go to the Jackson website and you can look up a lot of information on that. So basically, the first transgenic mouse line available through the Jackson Lab, it's a transgenic mouse line, and it's driven by an artificial, different promoter, which is K18. And the second, the 2 humanized human ACE2 model 1 and model 2, actually the human ACE2 gene is expressed and controlled by the endogenous mouse ACE2 promoter and its regulatory element. So at the end of March 2000, this is 2020. Dr. Ira Taps contacted me to work on a mouse model with the instrumental input on the design from Jahar and the funding through Dr. Don Landry's professorship. So we decided to generate a master mouse line that can be induced to express the SARS-CoV or SARS-CoV-2 receptor specifically in specific tissue. So we decided to knock in a CAC promoter stop cassette and human ACE2 tagged with C9 tag and IRES eGFP gene cassette into the mouse ROSA26 locus. And so in the early April, we met the gene targeting vector. And at the end of April, we have targeted ES cell. We did this project in the B6 and ES cell. And because it's still under the lockdown, so we were not able to perform the blastocyst injection to generate the chimera. So at the end of June, we actually, after the reopening, we were able to perform the blastocyst injection to generate the germline chimera. And we, at the end of August, we got the germline chimera were made. And at the end of November, we confirmed the germline transmission of this modified allele. So here I show you two master mouse line that in the A mouse line, actually, we knock in the synthetic CAC promoter with a flat stop cassette, followed by a human ACE2 C9 open reading frame and IRES eGFP. So we introduce this cassette into the ROSA26 locus. So ROSA26 actually is a well-known safe harbor. So any gene got knocked into that safe harbor site would have a very high tendency to express at high level. Sorry. And so we have the A mouse line that has the stop cassette, human ACE2 and IRES eGFP. And after we have this, the A mouse line, actually I went ahead and crossed A mouse line to a fleet-paste deleter to remove the IRES eGFP to generate the B mouse line that has the ROSA26 CAC promoter, stop cassette, and ACE2C9 without eGFP. So my purpose is that maybe some lab may not need the eGFP. So since we have the master mouse line A already, so why not generate a master mouse line B so that people will have more choice. And then for the purpose for this design is to be used for tissue-specific expression of human ACE2 in mouse. So this mouse line actually is different from those three mouse line available from Jackson Lab. Those three mouse line actually they will be the human ACE2 will be expressed either in the epithelial cell or in all the cell type that the ACE2 promoter is active. But in this new mouse line, we introduce like artificial promoter with a stop cassette into a safe harbor. So if we cross this master mouse line to a tissue specific tree, then we will be able to express the SARS-CoV-2 receptor specifically. I mean, in a tissue specific manner. And also in order to, we generate this mouse line, but we have not confirmed the protein expression in this mouse line. So in order to do it, actually I cross the master mouse line with the stop cassette to the E2A cre mouse, so that the stop cassette would be removed, so that the human ACE2 C9 open reading frame will be expressed from the CAC promoter. So what I'm trying to do is in the coming couple weeks, I'm going to get the service from the pathology core to do some immunohistochemistry study just to prove that this protein indeed is expressed. But actually this is a little bit concerned for this mouse line, because actually this mouse line is essentially a carrier for SARS-CoV-2 virus. So to maintain this mouse line, there's a certain risk, and maybe we need a certain protocol to maintain this mouse line. So so I'm in the process of talking to the ICM to see how we should handle this the ubiquitous the mouse line with the ubiquitous expression of human ACE2. But for the master mouse line A and B, I think it should be okay, because we have engineered a stop cassette, transcriptional stop cassette, to stop the expression of human ACE2 from the CAC promoter. So we have all three mouse lines available from our core facility for the time being. So the model A would be with the CAC promoter stop cassette, a human ACE2 and IIS eGFP. But I have to emphasize that actually this IIS is not that efficient. So the eGFP expression may not be that high. And the B master mouse line would be very similar to the A mouse line, except that the IIS eGFP has been removed. And the C mouse line actually is a ubiquitous, the HACE2 ubiquitous expression mouse line. But I think the C mouse line essentially is pretty similar to the three, actually, at least the two humanized mouse line available through Jackson Lab. So the C mouse line, I generate this C mouse line is because I need to remove the stop cassette in order to do the immunohistochemistry to verify that the introduced human ACE2 protein is indeed expressed in this master mouse line. So now the availability of this three mouse line would be, for the A mouse line, we have like limited quantity, but we are expanding this colony. So it would be, for the timing it's limited, but it would be available to all the Columbia researchers maybe at the end of February. And the B mouse line is also available. It's limited for the time being, but it would be readily available at the end of February. And the C mouse line, because we crossed the A mouse line to E2A cre, so the F1 generation actually is mosaic, even though we can show that the stop cassette is removed, but it's not homogeneous. So we need to go through another generation to fix the genotype. I mean the removal of the stop cassette. So it would take longer. But I think the C mouse line, if you are interested in the C mouse line, you may just go ahead and order the humanized mouse line from Jackson Lab, because I believe that essentially they would work the same. So if any group are interested in any of the mouse line, I would say please email or phone us, so that we would have a rough idea in terms of the how many groups are interested in the new mouse line, so that we can expand this mouse line and make them available accordingly. So my talk is really simple. So basically, I just want to let you know that we during the lockdown, actually, in the past year, we generate like a new mouse line that can be used to express the SARS-CoV-2 receptor in a tissue-specific manner. So this is a new reagent available to the research community. Okay, so that's it.
Yeah. Thank you, Victor. your talk went a little under time, so there's plenty of time for questions if anyone from the audience would like to raise their hands. If not, I'm going to go ahead and start off with a quick question, and that is, so if I understood correctly, mouse line C has ubiquitous expression of the human ACE2 receptor, correct?
Yes.
Can you comment on the levels of expression and whether those levels change in different tissues?
Okay, so it's driven by the synthetic KEC promoter.
Mm-hmm.
Okay, so the KEC promoter actually is a very robust promoter. But I do believe that the KEC promoter activity would also varies among tissue. Okay, but I don't have a exact answer to your question because I have never really, spent time to analyze the activity of the KEC promoter in different tissue. But the KEC promoter is so far considered to be a pretty strong promoter. And the reason why we use the KEC promoter instead of using the endogenous ROSA26 promoter is that we noticed that the ROSA26 promoter itself is not active in the retinal neuron. So it may also not be active in some CNS neuron. So that's our concern. So that is the reason why we did not use the endogenous ROSA26 promoter for this knocking. Instead, we use a synthetic KEC promoter.
Okay. We have a question from a panelist, Jeff Buka. So go ahead, Jeff.
Yeah. So my understanding is that the mice that ubiquitously express ACE2 basically die after you infect them with the coronavirus. Can you talk a little bit about like how long that takes and what they die of and what's been learned from that? Because my understanding is it's not considered a good model for the human disease.
Yeah, actually, I have to apologize, because I made this mouse model. I do not work on the COVID-19 research. So the animal model that you mentioned, actually, it will make the animal susceptible to the SARS virus or SARS-CoV-2 virus. And so how quickly they would die, I would say, it really depends on the infectivity of the virus or the condition, or maybe also the age of the animal. And that, because they would die during the infection. So that is the reason why we generate another, this is not an inducible one, but we do it in a tissue specific manner to express the human ACE2. And just in one particular tissue, not in whole body. So this novel model may be a little bit better to study the pathology of the COVID-19 in specific tissue. That means that the mice may not be dead right after the infection of the virus. So, I mean, we generate this mouse line, we just want to provide the research community another option of an animal model, so that if this animal model can be useful, then we would be very grateful that actually it may help people to advance the like study of the COVID-19 or even come up with a better treatment for the COVID-19. But this is a new model. We don't know. We want to make this model available to all of the Columbia research community, so that they would be able to generate a lot of data using this model. But I make this animal model, I do not study the COVID-19, so I don't have a good answer to your question. Sorry.
All right, we have a question from Jahar Bhattacharya. Jahar, you can talk if you unmute yourself.
Okay. Thanks. Yeah, I just wanted to address that very important question that was just raised and very nicely answered by Victor. The reason why Ira and later I got involved with making this model with Victor, not making, but getting involved with the project, was that we wanted a definitive expression of the human ACE2 receptor on the alveolar epithelium. And the reason we wanted that is because the alveolar epithelium is the oxygenation interface of the lung. And when the alveolar epithelium gets damaged by viral infection, there is pulmonary edema and therefore ARDS due to COVID-19, which causes death. Now, there is no mouse model that convincingly causes this receptor expression on the alveolar epithelium. The K18 model that the commercially available JAX models do not express the receptor on the alveolar epithelium. The K18 promoter, for example, is a cytokeratin 18 promoter, which does not even exist in the alveolar epithelium. So all of the expression happens in the airway. Sure, mice die, but they die because of other reasons than ARDS. They die of cerebral infection. They die of, CNS infection and airway obstruction, all kinds of reasons that are not relevant to human disease, the human COVID-19. So we wanted to see whether it's expressed. All right, now the point about is the mouse model good for studying COVID-19 ARDS? That's a great question. And my answer is there is no mouse model that exactly replicates any human disease. And yet we have understood human disease by studying mouse models. There are any number of examples of that, diabetes, for example. So what we can do with this mouse model that Victor has so nicely developed is we can study signaling. We can study the way the alveolar epithelial cells, after getting infected by coronavirus 2, actually signal amongst themselves, because we think the breakdown of the barrier is a multicellular event, multicellular mechanism that needs to be understood at the level of cellular organization in the alveolus so that we can come up with therapy. At this point in time, we just are not able to touch this problem because there are no models, mouse models or otherwise, by which we can even address this issue.